These molecular epidemiological techniques, or DNA fingerprinting techniques, have a wide variety of acronyms, and it's somewhat complicated to the uninitiated. So PFGE, or pulse field gel electrophoresis, this was the method we used in the E. coli and chicken example. We have AFLP, or amplified fragment length polymorphism, RFLP, restriction fragment length polymorphism, variable number tandem repeats, multiple variable number tandem repeats, or random amplification of polymorphic DNA. These are all what we call band-based techniques. They indirectly look at polymorphisms within the genome without actually sequencing it. So it's based on digesting the DNA with restriction enzymes and amplification using PCR. On the right, we have our sequence-based techniques. We can either use universal targets, something like 16S or CPN60. We can have more hypervariable techniques, perhaps a gene like SPA, which is used for typing methicillin-resistant Staph aureus. Or we can have multi-locus techniques. So multi-locus sequence typing involves sequencing multiple housekeeping genes and comparing the sequences of those to a database. And then finally, and the techniques that are really sort of taking over molecular epidemiology, are whole genome sequencing. And this can be based on a simple amplification of DNA, as we see with the Oxford nanopore technology, or sequencing digested fragments of whole genomes, as we saw with older technologies. With all this alphabet soup of acronyms, how do you choose which method is best? In my opinion, there's really no such thing as a good test or a bad test. There's just a test which is better or worse suited to addressing the particular question you're interested in. And when we consider our molecular epidemiological techniques, I think it's useful to think about them in terms of their resolving power. And what we mean by resolving power is how similar can two isolates be to each other and still be recognized as different. So techniques like sequencing universal targets are maybe good for differentiating the genus or species, maybe strain. As we move up through some of these other methods, MLVA, PFGE, MLVA, we start to be able to differentiate closer and closer relatives to each other. Finally, whole genome sequencing is theoretically the most discriminatory. A single nucleotide difference between isolates will identify them as different. Another way to think about this is in terms of sensitivity and specificity. So techniques like universal target sequencing will be relatively insensitive to identifying differences between closely related strains. The flip side of this is specificity. So whole genome sequencing can actually identify differences between strains which are not actually epidemiologically relevant. The molecular clock of the organism, or the rate at which single nucleotide polymorphisms occur, can actually be so fast that we start to see variability within the course of an outbreak. And so it takes specialized knowledge and expertise in order to essentially discount some of these differences. We're going to discuss a couple of these methods in a little bit more detail, starting with pulse field gel electrophoresis. This is a method which I think is rapidly becoming obsolete with the widespread availability of whole genome sequencing, but it's historically very, very important and is still certainly quite useful. The way that we perform this method is we start with a pure culture on agar, we suspend some of those colonies in broth, and then incorporate that colony suspension into an agar plug. So we actually take molten agarose and mix that with our bacteria. We then lyse the bacteria within that agar plug using buffers and detergents. We then digest the genomic DNA into pieces using restriction enzymes. And then we resolve these DNA fragments on a gel using a specialized electrophoresis apparatus. So this is what you can see here is the pulse field gel electrophoresis chamber. And the way that this works is we have this pulsing of the current that moves from side to side and allows us to separate very large molecular weight DNA fragments up to megabases. Interpreting pulse field gel electrophoresis data involves comparing the banding patterns of individual isolates to each other. So in this gel here, you can see we have 4 control lanes This is a known organism which has a very particular banding pattern that we can use to normalize the size of our bands in other planes. And then what we do is we compare the isolates to each other. So these three on the left here, you can see have the same banding pattern and would be considered to be indistinguishable by this technique and therefore related to each other as compared to some of these other strains. Multi-locus sequence typing is another commonly used DNA fingerprinting technique. In this method, again, we start with a pure culture on blood agar. We do a DNA extraction on that culture, and then we do a PCR for 7 different housekeeping genes. So these are loci which are known to be conserved within a species or genus of bacteria and evolve at a relatively slow and consistent rate. Once we've done that PCR, we then sequence those 7 amplicons and use some sort of web-based interface and database in order to compare the gene sequences for each of those 7 genes to what's been found previously, and then subsequently identify the sequence type. So that was a long explanation. What does it actually look like? This is a screenshot of Enterobase, and here you can see the E. coli and Shigella MLST platform. There's a wide variety of other organisms for which we have MLST schemes published for, not just within Enterobase, but there's other web-based platforms. Essentially, what we do is we take our DNA sequence from each of those 7 genes, paste it into their web form, search. The database assigns each of those sequences an allelic number. So each ADK sequence, for instance, is numbered 1, 2, 3, 4, 5, etc., in the order that that sequence was identified. And the overall profile of these 7 allelic numbers is what defines the sequence type. So here you can see some data on E. coli. What we have here are the allelic profiles, so 53, 40, 47, 13, 36, 28, 29, and this represents sequence type 131. MLST is a technique that's also kind of falling out of favor. Doing PCR and amplicon sequencing on these 7 housekeeping genes is actually almost as expensive as it is to sequence the entire genome. And so increasingly what we're seeing is MLST performed in silico on whole genome sequences, which are much more efficient to generate these days. So whole genome sequencing is obviously a very rapidly developing technology. In some ways, I think it's moving faster than our ability to meaningfully use it. It's sort of a hammer looking for nails. And we need to be careful of sort of the answer box syndrome, thinking we can just sequence everything and get a quick and easy answer. I think we're getting there, but it's going to take a bit more time, and expertise is still really required to meaningfully analyze this data. One of the most important things is posing a question that your tool can actually address. So what do you actually want to learn by sequencing a genome? Interpretation is still really the main limiting factor, and so having a good question to start with is really important. As I said, whole genome sequencing is starting to replace all of these classical techniques. And so things like pulse field are rapidly finding their way into the microbiology history books. The cost of whole genome sequencing is rapidly going down, and it's being increasingly adopted by major organizations like the Public Health Agency of Canada, Public Health England, the United States FDA, and it's being used widely in outbreak investigations and sort of general molecular epidemiology applications. When we're talking about whole genome sequencing, what we're really talking about is what used to be called next generation sequencing. It's not really accurate anymore. We're kind of into the 3rd generation of DNA sequencing technology. What we're really referring to is massively parallel sequencing. So the ability to sequence many, many, many DNA fragments simultaneously, which allows us to essentially sequence the entire chromosome all at once. So just as an example, something like Illumina would allow us to sequence 10 to the 4 to 10 to the 9 DNA fragments simultaneously. So up to 1 billion. And if you consider that they can be hundreds of base pairs long each, we're looking at hundreds of billions to even trillions of base pairs worth of data in a single sequencing run. Now, this compares to Sanger sequencing. classical sequencing technology, which could handle at most about 384 fragments simultaneously, so orders of magnitude less. This sequencing technology, the chemistries, and the computing power is in fact advancing so rapidly that our first generation, next generation technologies are already becoming obsolete. So 454 pyrosequencing, introduced in 2005, was sort of the first of these massively parallel sequencing technologies, and it's been completely retired since 2016. At present, platforms like IonTorrent, Illumina, PacBio, and increasingly Oxford Nanopore are used for whole genome sequencing, each with its own set of advantages and disadvantages. I'm not going to go into the specifics of the sequencing chemistry and sort of how each of these work in this lecture. It's sort of beyond the scope of this class. So when are these methods used? When do we need to do DNA fingerprinting? Well, ultimately, the goal is source attribution. So if we're doing an outbreak investigation, this could be at a national or international level. Maybe there's a food product that was contaminated with salmonella or E. coli, and we need to identify where that's come from. This could also be within a healthcare facility. Maybe there's a nosocomial outbreak of an infection linked to a medical device, for instance. We can track the movement of strains within the community, whether that's in animals, people, the environment. We can use these methods to study bacterial evolution or to monitor the dissemination of mobile genetic elements, those plasmids that are so frustrating, particularly in the gram-negative world. Many countries, including Canada, have national level organizations like the Public Health Agency, which utilize these methods in ongoing surveillance, whether it's of diseases and outbreaks, or for looking at potential pathogens in a prospective way. So doing things like looking for resistant organisms in retail meat. Big picture messages today. Molecular microbiology is not magic. There is no one technique that will solve all of our problems. And so some serious consideration is really required in order to determine which test is best suited to a given clinical situation. If you think that you're dealing with an outbreak, if you see multiple cases of infection with potentially the same organism, talk to your diagnostic lab. They can help you to determine what the next best diagnostic course of action is, what other tests need to be run, and can put you in touch with someone who can help you. And finally, at least in a bacterial context, avoid the temptation to infer relatedness and presumably some common source based on antimicrobial susceptibility alone. There's that homogenizing pressure of local use, and remember, common susceptibility profiles occur commonly, so we can coincidentally see the same thing in unrelated cases of infection. And finally, 2 questions for self-assessment.